the screen. And putting it up, and then it didn't. Wait a minute. Somehow it popped right back to the end again. There we go. Okay, now we're ready. So the title of the talk is the coronavirus and its relationship to an enzyme that's associated throughout the entire kingdom of life with toxicity, phospholipase A2. And so the coronavirus uses phospholipase activity to infect, replicate, and harm its host, but it is a target for perhaps some simple nutritional supplements. So I'm going to be talking about that. And the outline is I'm going to start with what phospholipase A2 is and why it's so important and how it damages living things. I'm then going to focus on the coronavirus, a brief overview of the epidemic. I'm going to then turn to how the viruses are so involved in phospholipase A2. And I'm going to talk about a nutritional supplement that is a potential antidote and for phospholipase A2. I'm also going to talk about other diseases unrelated to viruses that are also mediated through excessive phospholipase A2, and those include heart disease, stroke, multiple sclerosis, Parkinson's disease, Alzheimer's, and memory loss. And there's many more than this, but that's just a sample of the diseases in which phospholipase A2 plays a role.
Dr. Shah, can you maximize your screen?
Yes, I can. There we go.
Thank you.
There we go.
Perfect. Okay.
Thank you. So a wide range of creatures that are toxic or venomous transmit their toxicity in their venom by activating phospholipase A2. The poisonous snakes, like this snake, and also rattlesnakes, the black widow spider, and wasp, the stinger, the venom from the bee. The pain you feel is when a wasp or a bee stings you is because the phospholipase A2 is being injected into your skin, which is then producing what I call biological detergents, which can be very painful. So the products of phospholipase A2 cause inflammation and pain. The bacteria, such as E. coli, it infects the cells and causes harm to the host by producing phospholipase A2. And parasites, such as toxoplasma, an organism that is harbored in cats and can cause severe illness in humans, also infects humans by secreting phospholipase A2 to burrow into the human host. The coronavirus, and in this case, the coronavirus 19 is likely produces phospholipase A2. All the coronaviruses do that, and since it's closely related, it's very likely that it produces the phospholipase A2 also. The The phospholipase A2 is produced by microbes, which include viruses, bacteria, and yeast, and is one of the major factors that's associated with human pathogenicity and death. A condition called sepsis is an extreme infection that's frequently fatal, caused by an overgrowth of microorganisms. And there's very good evidence that sepsis itself is due to the overproduction of phospholipase A2. And it's one of the major factors involved in venom of snakes, spiders, and stinging insects like bees and wasps. Phospholipase A2, when it's elevated, can also be produced by humans directly, and it is a major factor in autism, Alzheimer's, multiple sclerosis, atherosclerosis, pancreatitis, which is inflammation of the pancreas, many types of cancer, depression, schizophrenia, and probably many other diseases. The mechanism of why phospholipase A2 is so toxic is one of my lifetime interests. So I started doing research on the phospholipase A2 while working at CDC in Atlanta. And the reason was, at that time, it was found, excuse me, I'm gonna have to stop for water for just one second. So what I found was, at that time, people were doing tests for thyroid hormones, and it was found that all thyroid hormones throughout the world were giving incorrect values. And this would have been a giant to-do if there was an internet at that time, because all people who were sending in their thyroid tests were thinking they were getting accurate results. But for an extended period of time, there was no accurate results throughout the entire world because the thyroid hormone tests were completely unreliable. And so my job was given to find out why. And what I found out is because of the phospholipase producing these broken down fats that were inactivating the proteins in the patient samples, so that it gave inaccurate results. So this was a major scientific achievement, but of course at that time the only people who knew about it were the other people in the laboratories who read the papers. And so I went from that to thinking, what if the same thing happened, this elevation of so the phospholipase was causing inaccurate results when it was forming in blood tubes, but I thought, what if it happened inside a person's body? And so I connected a number of different diseases caused by elevated phospholipase 2. One was a disease of children, which was commonly fatal, sudden infant death syndrome or crib death, pancreatitis, and diabetic ketoacidosis. And this is just one of the examples. So Reye's syndrome is a disorder of children usually following chickenpox or flu that results in nausea, lethargy, coma, and death. There's frequently increased intracranial pressure, abnormal EEGs, and when they analyzed the blood samples of children with Reye's syndrome, they found that they had abnormal fats that were indicated likely overactivation of phospholipase A2. And at that time, aspirin was initially suspected as a factor, but later studies cast doubt on that hypothesis. And even though later studies indicated that there's a good chance it wasn't aspirin, this was the death of baby aspirin because of the suggestion. It was the death of baby aspirin and a marked increase in substituting baby aspirin with Tylenol. And then as Tylenol changed over, then this was associated with the large increase in the incidence of autism. And you can see some of these webinars on the autism epidemic and its connection with Tylenol are listed on the website under the archived webinars. I also want to tell people you can write in questions at any time. I'm not going to answer them until the end of the webinar, but you can you can type in questions at any time, and then at the end I'll be answering quite a few, and it'll probably be till my voice gives out. So that'll be the so I'll be glad to answer questions. So in 1985, when I published the paper about phospholipase A2 being involved with Reye syndrome and crib death, and there were no hospital or commercial laboratories performing any phospholipase A2. And now in 2020, there's the Great Plains Laboratory is the only laboratory in the world performing phospholipase A2 on urine, which is a convenient sample. Some commercial laboratories analyze one subtype of phospholipase A2 associated with heart disease in blood, but they're not measuring the phospholipase A2 that's involved with viruses. And the lack of testing is probably the major reason why there has not been adequate research about the role of phospholipase A2 in causing pain, its pathogenicity, causing disease, and causing death. So there's probably millions of laboratories throughout the world who test cholesterol, but as far as the phospholipase A2 in urine, they're just one. And so what I've seen is the availability of testing is a major thing that draws attention to researchers to look at different topics. And I'm convinced that if there had been a readily available test, you would find hundreds of thousands of articles and many, many drugs that were already in the market for controlling phospholipase A2. So this is the molecule that's broken down by phospholipase A2, and it's called lecithin. And if you look on the back of read the ingredients on your Hershey bar, you'll see that a lot of time that is listed as soy lecithin commonly. Another name is phosphatidylcholine. And so you need to know this particular name. So this is the structure. It's written as two ways. And on the left, the phosphatidylcholine is written as just a two-dimensional structure. On the right, it's written as a three-dimensional structure. And it looks a little bit like the old-fashioned clothespins that you would use for hanging clothes on a rope outside the house. So the extensions of the clothespin here are oil soluble. They're called hydrophobic, meaning they don't like water. And so they're oil or fat soluble. And there's two of these present. And on the right side is showing the same thing, except it's showing it as a 3D image. Here at the top of the lysolecithin is the hydrophilic or water-loving part of the molecule. And the reason, and it's more water-soluble, and it's because it has charges on a positive charge here and a negative charge on the phosphate. So having both the positive and negative charge makes this part of the molecule very water-soluble. Phospholipase A2, what it's doing is breaking down lecithin, which is a weak detergent. So you may think, well, what are you talking about detergents? Are we talking about somebody's laundry here or something? But that's exactly an appropriate analogy. So it has the same, if a thing is a detergent, it means it has the same kind of abilities that your Tide would have in the washing machine. But lecithin is a very weak detergent, and phospholipase A2 removes this fatty acid in this particular portion, and it breaks it down into what is called a litholecithin, which is a very strong detergent. And also the most common lecithin has arachidonic acid as the fatty acid, and arachidonic acid is also a strong detergent. So phospholipase A2 converts a very weak detergent into two very powerful detergents. And this is a typical article. I mean, there's hundreds of these out there. Lysolecithin, which is also called lysophosphatidyl-choline, induces pain. So that is one of the big things that's causing pain, and I think that's what the reason for why there's a lot of pain going on in the coronavirus infection is because the virus is causing a lot of production of this lysolecithin. So I want to show what the biological detergents are. So I'm comparing soap. So ivory is you can see here on the ad, 99.44% pure. And ivory is made up of a fatty acid that's present in your body and around your waist called lauric acid. Lauric acid has 12 carbons. You start down here, here's number 1, 2, down to the end, there's 12. And fatty acids like this can be converted to soap. And so that would be the main constituent of ivory soap. If you had Tide or any number of other laundry detergents, they also have 12 carbons, but at the end they have a sulfur group that makes them, it makes them able to work even in a non-ideal environment like hard water, for me, highly mineralized water. So the name of that is very similar to the fatty acid that's found in ivory, lauric acid. In Tide, it's lauryl sulfonic acid, but again, it has 12 carbons. So these things that you use to wash your hands these days and that you use for cleaning your clothes are detergents, but phospholipase A2 has the ability to produce detergents that are similar in potency to these laundry detergents. So these biological detergents denature proteins so they don't function very well, or sometimes they don't function at all. And like the laundry detergent, they have a water-soluble and a fat-soluble end. And the two major biological detergents are lysolecithins and free fatty acids. And one molecule of lecithin the other name of which is phosphatidylcholine, produces two molecules of very powerful detergents, lysolecithin and arachidonic acids, which decrease surface tension. So that you can, if you put a pin on still water, sometimes it will float due to the surface tension, but if you add a little bit of detergent to it, it will sink. So the reason these biological detergents are so important is because all proteins have a three-dimensional structure. I know to many people this looks kind of chaotic, but almost every protein has a structure, or many proteins have a structure very similar to this. And these are not random. They, in other words, these are, each molecule that's made will have almost the same structure. And detergents react with those and convert it to just a random string. And that random string then has no function. It can't work as an enzyme. It can't work as a building block in the cell. So there's, maybe 20,000 different proteins in the human body. And virtually all of these would unwind and lose their ability to function when detergents react with them. And when I first discovered this phenomena that was causing the breakdown of a protein that binds thyroid hormone, what I found is that each of these fatty acids has a different detergent potency. So saturated fat had little, almost no detergent ability, but arachidonic acid, one of the largest unsaturated fatty acids, was the most potent biological detergent. So each of the fatty acids, which occur in blood and throughout your body, in your fat and so forth, these free fatty acids have extreme detergent activity. And it's this particular fatty acid, the one that has this powerful detergent, that's released by phospholipase A2. So now I want to deal with the coronavirus, and it's called COVID-19, or another name it's given is severe acute respiratory syndrome coronavirus 2, which is abbreviated as SARS-CoV-2. The coronavirus has these characteristic spikes, and somebody had the idea that this looks like the sun when there's an eclipse, that you will sometimes see the flares from the sun sticking out when there's a total eclipse. And so that was the reason for calling it the coronavirus. All the coronaviruses have this type of spikes that look like solar flares. The coronavirus, as you all know, now has spread throughout the world. The darker the red color, the higher the incidence of the coronavirus. And you see here, Italy is one of the most infected areas in the world. But you also see the United States is getting it, the west coast of South America, Peru and Chile are also getting it. And this is very important because these countries are just getting over summer. So the idea is that this virus is not going to persist in the summer, I think is ruled out by the fact that these countries were right in the middle of summer when the coronavirus landed there, and now they have their rates of infection are very high. And the same thing with the Australia. So in the southern hemisphere, the summer was just being completed, and so the virus really took hold during the beginning of summer, and which there is January. This is the structure from Wikipedia, and here's the spike showing it has a carbohydrates attached to it, sticking out, and then the coronavirus. Almost all living things use DNA as its genetic material. The exception is the coronavirus, which has a RNA. So in this case, it is a single-stranded. As many of you know, the DNA is a double-stranded molecule. The RNA of the virus is a single strand. And a pretty disgusting picture. So this is why this is the reason why you want to be always at least 6 feet away. As a matter of fact, looking at this, I would say maybe you would need to be 12 feet away from this guy. The source of the coronavirus in China is almost surely due to what is called the wet markets. The wet markets just meant that you could get almost any living creature. So when you look at the types of animals that are eaten in China, it's a very long list. And some of these I had never heard of. So the thought is that it originated, and all these animals are live animals that were in cages where people would buy them. And I don't know if they would slaughter them on the spot or you would take these animals live and then kill them at home. But it's thought that this almost surely was the thing that caused the production of a mutant coronavirus from bats that may have transmitted the virus to an animal called a pangolin, which I had never heard of before, I don't think, and that the person eating the pangolin then became infected and then spread it to all the neighbors. So like the coronavirus, a wide range of viruses that attack humans use phospholipase A2 to get inside your body and attack it and damage it, and so that the virus replicates. So hepatitis C, you've seen lots of ads on TV for drugs that treat hepatitis C. It is a single-stranded RNA, just like the coronavirus, and it replicates inside the liver cells of the host, and one of the proteins produced by the virus hijacks the cells it infects and causes the cells in the liver to start producing excessive phospholipase A2. And if the gene for phospholipase A2 is removed from these cells, the infection rate of the hepatitis C drops dramatically by putting them into cells that don't have phospholipase A2. You can also use a phospholipase A2 inhibitor, and it also causes significant reduction in the hepatitis C virus replication. The influenza virus also causes infection by activating phospholipase A2, and the the phospholipase A2 breaks down the lecithins and produces the lysolecithins, which alter the plasma membrane and the membrane apparatus of the cells, and causes it to form a shape that is more conducive for the virus replication. And so the article summarizes that the PLA2 may provide a therapeutic target. In other words, this is the vulnerable point of a lot of viruses, and so this may be a way to stop it. Another virus called CMV stands for cytomegalovirus, and in this case, the virus actually carries along phospholipase A2 and the RNA of human cells that can make more phospholipase A2. So this case, instead of just hijacking, this has made the phospholipase A2 of the human cells a hostage, which then has to travel with it and helps it to infect other cells. So, so the combinations of inhibitors of this, again, could be used for treating another infection. So a wide range of viruses replicate and hijack or make hostage of the cells, either taking their phospholipase A2 and including it, taking it along with it, or altering in the early stages of the cell, altering so the cell produces excessive phospholipase A2. And also a parvovirus is another virus, I think is more involved with dogs, also contains phospholipase A2, in which the virus itself is carrying its own phospholipase. So here's an article, the American Society for Microbiology Virology Journal, that phospholipase A2 is an important step in the coronavirus replication and cell culture. And it's doing basically something very similar to the other viruses. And it produces the breakdown of the structure of the cell, converts the structure to vesicles that the virus can insert itself in and has a greater ability to replicate. And so all of these things are being altered. They're altering the fats, the lipids inside the human cells, and by using electron microscopy, they found that if you add phospholipase A2 inhibitors, you significantly decrease the presence of these abnormal structures that are produced by the virus to help it replicate. So by using inhibitors of phospholipase A2, you're able to appreciably decrease the replication of the virus. And the particular inhibitor they used in this particular study was called pyrrolidine. And pyrrolidine, to my knowledge, has not been used as a nutritional supplement. I think it is one that might be used at some point for research, but I couldn't find any articles. It's a small molecule, but it's a very potent inhibitor of phospholipase A2. And it was also knocked out the phospholipase A2 and the virus activity of other viruses as well. And so the conclusion of the study is that these inhibitors of phospholipase A2 may represent a candidate target for broad spectrum antiviral drug development. And here is a slide showing the amount of virus. The taller the bar, the greater the amount of virus being produced. And this is going to me a little bit backwards. So it's to say that if you don't add any, if you put the virus, you take the coronavirus and put it in cell culture, this is how much virus is produced if you don't have any phospholipase A2 inhibitor. And then if you increase it, it drops down. And then, say, here's the concentration, 0, 5, 10, 20. So going from 0 to 5, it drops a little. Doesn't drop too much going to 10. It's almost the same. But when you hit 20, so you see there's 2 stars, 2 asterisks. That means it's a very significant change. It means this is only a 1 in 100 chance of being probability of being due to chance. So you went from 0 to 5 to 10. When you hit 20, the amount of this pyrrolidine, it really dropped the virus from the titer was 100,000 to 1,000. So it 99% reduction in virus replication by using this inhibitor of phospholipase A2. And the Ha up here is just the name of the human cell culture. And so they tried 3 different cell cultures, and they found that the Ha and the BAS were extremely inhibited. The MRC cell culture wasn't quite as inhibited as the other 2. And then here is the timeline. The solid line up here is the virus replicating without any inhibitor and no inhibitor of PLA2. And then as you increase it, the virus drops and drops over 24 hours. So roughly it was a 10 to the 6th to 10 to the 4th. So again, you know, it doesn't look as dramatic, but this is really about a 99% direction because this is like a logarithmic scale. So the amount of virus decrease was about a 99% reduction with the higher amount of the pyrrolidine. And this is looking at the cells under the microscope, and in these slides, the red dye is the virus protein. So they add an antibody that stains the coronavirus. So a red color means the cells in the cell culture are infected with the coronavirus. So here are some in which cells that don't, these are the normal cells that don't have any virus. And then these are the infected cells with no phospholipase A2 inhibitor. And then here's the same cells that were infected with just as much coronavirus, but they added the phospholipase A2 inhibitor. So none of the red stuff is there, meaning there's no infection or almost none. There, I guess there could still be some, but there's very little. It's not visibly demonstrated like it is in the cells that are infected with the virus, but are not given the phospholipase A2 inhibitor. And Again, this is something that, is very similar to the current coronavirus, and it's called the Middle East respiratory virus coronavirus, but it was one that was associated, spread from camels, and it became very potent in the Mideast, and the fatality rate became extremely high with that particular coronavirus, up to 36% fatality with people who were infected with this coronavirus. And this is a good thing. This is a good graph showing that all the different cells, the virus was decreased as you. So if you just look at this solid bar here. Look, let me go back. Went ahead. Look at the cells that don't have any filling. So it's this one, this one, this one, and this one. And then as you increase the amount of the phospholipase A2 inhibitor, it declines and declines significantly. So it is about 1,000-fold less after treating it with the phospholipase A2 inhibitor. This is another coronavirus, and these are some other types of viruses. This one, the rhinovirus, it had some effect, but you see probably one of the biggest effect was on this this virus that was very similar to the present epidemic coronavirus. And so again, this is one cell culture, significant drop, and this is another cell culture with the hatching in the bar. It dropped and dropped and dropped again. In this case, it dropped again about 1,000-fold. Now, whereas influenza virus, it wasn't any good. It didn't. Influenza virus had very little effect. Polio virus was not helped very much by inhibiting the phospholipase A2. It was mainly the coronavirus. And so another study looked at the specific change in the fats or lipids after being infected with the virus. And what it shows, so we look at this graph here at the top, this figure 2, number A, here on this axis, this is showing the amount of increase compared to a non-infected cell. So after the coronavirus, the toxic detergent lysolecithin, this particular one was about 7 times the amount of cells that were not infected. The cells were not infected. And each one of these bars is a separate lysolecithin. All of them are increased. And these are all toxic detergents that can destroy the structure of cell, that can change the structure of the cell, and in fact, hijack it so it's in a format that's more amenable to making replicas of the coronavirus. It's also found that after the infection, the most destructive fatty acid, which is arachidonic acid, the fatty acid with by far the most potent detergent activity, has increased about 7-fold after the coronavirus infection, showing that the coronavirus is breaking down all the normal fats, which are called lipids, in the cells and converting them to toxic detergents. Another detergent is called, it's very similar to lysolecithin, and it's called lysophosphatidylethynolamine. It has very similar characteristics, and it is also increased following the virus infection. And then if you look at the fatty acids themselves, remember that phospholipase A2 produces 2 detergents for each molecule of lecithin that it breaks down. One is lysolecithin, the second is free fatty acids. So when they look at the free fatty acids, they were also markedly increased, with one of the biggest increases being the most toxic fatty acid, arachidonic acid. So the summary is a number of different viruses, including the coronavirus, infect the cells and injure them by producing their own PLA2 or hijacking the PLA2 of the host cells to produce PLA2. The excessive PLA2 produce excessive biological detergents such as lysolecithins, lysophosphatidylethanolamine, which have the ability to break down the structure of the cells. The biological detergents are formed throughout the body, and that's why this current coronavirus is so bad, because it's not just infecting some cells of the body, it's infecting a substantial portion. And these detergents, even if the virus isn't in there, the detergents can then move through the bloodstream and cause toxicity to all the other organs in the body. And if you inhibit the PLA2, it causes a marked viral replication, including the coronavirus. And there's a wide varieties of phospholipase A2, and most of them are relatively small as far as proteins. So this line right here is the molecular weight, and the molecular weight is a small molecule is any molecule less than 1,000 kilodaltons, and a very large protein could be over 100 kilodaltons. And most of the phospholipases in the body are relatively small molecules. And the reason this is significant is because the small molecules all appear in the urine, and most of them do not appear in the blood because they're all filtered out of the blood and appear in the urine. So the test at the Great Plains Laboratory largely checks these small molecules. So there's many different types. What is it, about 10 different types? The types that are measured by Quest and LabCorp is called lipoprotein-associated phospholipase A2. And again, these break down phospholipids, the lecithin, to form toxic detergents, and high amounts of this molecule are associated with heart disease. So the phospholipase A2 that is less than 15 kilodaltons, virtually all of it goes into the urine. For ones that are intermediate size, a lot of it, but maybe not 100%. And the ones that are above 45 kilodaltons, most of those stay in the blood. So now we get to the interesting part. So the interesting part is that there's a readily available supplement that's also been used as a pharmaceutical in Europe. So the name of the pharmaceutical in Europe, where I think it's still available. So those of you who are staying up late listening to this from Europe, it may still be available as a pharmaceutical. And the pharmaceutical name is Cytosolic, and the name used as from the supplements that are sold in the United States is CDP choline. And the name of this article indicates that Cytosolic or CDP choline is an inhibitor of phospholipase A2, and it also causes a reduction in toxic free radicals, which are called hydroxy radicals, which are some of the most toxic free radicals. So here is the structure of CDP choline. It consists of one of the bases of DNA and RNA, which is called cytosine. So every molecule of DNA and RNA has cytosine. So this is one of the 4 bases that make up the genetic code. It's the C of the ATCG genetic code. And it has the sugar ribose, 2 phosphates, and choline. So the name is, the full name would be cytosine diphosphocholine, perhaps. And the most commonly just abbreviated as CDP choline. So CDP choline is a strong inhibitor of phospholipase A2. And one of the most important things is you hear the word choline, you have to make sure you don't get these things mixed up. It could be fatal. So you better pay strict attention to this portion. Phosphatidylcholine is also commonly sold as a supplement, and it also has the name lecithin. So CDP choline is a strong inhibitor of phospholipase A2. Phosphatidylcholine or lecithin does not inhibit phospholipase A2. So when you go to your health food store, always read the labels very carefully. And if you think you may screw this up, please write it down and put it on a Post-it and stick it in your wallet. CDP choline decreases toxic lysolecithin and arachidonic acid, these 2 toxic detergents of PLA2. Phosphatidylcholine increases toxic lysolecithin and arachidonic acid if phospholipase A2 is there, because that's what it does. It breaks down phosphatidylcholine and produces toxic detergents. So you don't want, if you had the coronavirus, you definitely don't want to be taking phosphatidylcholine. CDP choline reduces inflammation and pain in a wide variety of illnesses. Phosphatidylcholine will increase inflammation and pain if you have high amounts of phospholipase, like you would from a virus infection. So this actually shows the reduction in phospholipase in animals that were made to have a stroke. And when a stroke happens, the amount of phospholipase A2 goes up. And so CDP choline has been useful for reducing the effects of stroke also. So this is looking at the cell membrane, and then on the right side, there's also PLA2 in the mitochondria. So let's look at this. So once the blood flow to the brain is stopped in these animals, the amount of phospholipase A2 went up, and it was significantly higher than animals that didn't have the blood flow cut down, which is simulating a stroke. And if they were given the CDP choline, all the animals had a smaller amount of phospholipase A2. So 45.6 versus 32, 48.2 versus 34.4. All of these were considered to be statistically significant at the very good probability level, less than 1 in 100 probability of this results being due to chance. And in the mitochondria, the changes due to CDP choline were even greater. 77 down to 41.9. So the summary of this article is that, so remember, these weren't viruses, these were, so there's different things that can cause phospholipase A2, and one was cutting off the blood flow to the brain. That was, so inflammation, lack of oxygen is also causing problems. So acetylcholine resulted in a decrease in PLA2 in both the cell membrane and the mitochondria. However, something that I didn't realize is that this is due to an indirect inhibition rather than the CDP choline reacting directly on the PLA2. It appears that it inhibits an earlier biochemical reaction, which ends up inhibiting the PLA2, not a direct inhibition. And the CDP choline or acetylcholine decreased these free radicals. These free radicals are very toxic, and so by getting rid of these free radicals, by lowering PLA2, it caused the mitochondria to be protected, functioning well, and producing energy, and also preventing arachidonic acid formation, which results in also producing reactive oxygen species and free radicals. So the what was interesting is that when you take CDP choline, it's broken down very fast, and it's believed that it's absorbed from the intestinal tract as two separate items, cytidine and choline. But if you give cytidine by itself, it's not effective at all on blocking PLA2. You need to give the combination. And so the combination caused neuroprotection where the cytidine by the C of CDP choline by itself was not effective. It's also interesting that it's been found a significant portion of people with multiple sclerosis, they found coronavirus detected in the brains of individuals who had passed with multiple sclerosis. And a second article finding the coronavirus nucleic acid sequences in the cerebrospinal fluid of patients with multiple sclerosis. And this is very interesting because we found high amounts of phospholipase A2 in patients with severe multiple sclerosis. You see here is the amount of phospholipase A2 in the urine, extremely high in people with severe multiple sclerosis, lower in people with moderate, and people with mild had the lowest amount of phospholipase A2, which was really close to normal values. We didn't publish our data, but other people published similar data in the Journal of Neuroinflammation and found exactly the same thing, that people with severe multiple sclerosis had the highest amount of phospholipase A2 in the urine. When people with multiple sclerosis were given CDP choline and then retested, we found a reduction of phospholipase A2 in the urine. So, what is the cause of elevated PLA2 in multiple sclerosis? Is it the effect of coronaviruses? So we don't know for sure, but it looks suspicious. Coronavirus is found in high amounts. Almost all the coronavirus producing excessive PLA2, and and a number of people found with MS had the cerebrospinal fluid with the virus nucleic acids. Alzheimer's is another disorder in which phospholipase A2 is elevated. This is a normal older people, 45 to 75 years old, and they have the amount of abnormal phospholipase A2 is the amount of gray area, whereas the people with Alzheimer's disease, looks like they have about 5 times as much. Even when they're young or they're older, they both have, people with Alzheimer's have high amounts of phospholipase A2, and several studies have come out to indicate that the symptoms of Alzheimer's were significantly reduced following the use of CDP-choline. We also found very high amounts of phospholipase A2 in children with autism spectrum disorder, ASD, and we've had reports of significant improvements in the autism symptom following CDP-choline. Another study in one of the journals found evidence of high amounts of phospholipase A2 in children with autism, and that by taking fish oil, the fatty acids EPA and DHA, they could it helped to reduce the amount of phospholipase A2 in autism. So from got this today from WebMD, and here's the URL where you can look this up yourself. It can be taken by mouth or given as an injection to help memory loss due to aging, improve vision in people with glaucoma, help with recovery and stroke. It's also used for Alzheimer's, Parkinson's, bipolar disorder. This is interesting also, lazy eye and other conditions of the brain. But there are not any gigantic studies because this is mainly a nutritional supplement. So there's, you can't sell this stuff for big bucks. So what about thinking about using it for coronavirus? No one has tried it yet, but PLA2 inhibitors in these cell cultures cause marked decrease in virus replication in the cell culture. However, all these biochemical studies indicate that coronaviruses and many other viruses infect cells in overproducing PLA2, causing high amounts of lysolecithins and fatty acids with extreme detergent properties that might be the cause of kidney and lung damage in coronavirus infection. So I looked at one of the reviews. She says, I've been taking 2,400 milligrams a day for 1.5 years. I find my thinking is clear, better able to focus, my memories improve greatly. It was recommended for my 85-year-old father by his neurosurgeon, and I asked if it would help me. And so she started at 600 milligrams and increased it up to 2,400 milligrams. The most common side effects are some GI disturbances and occasional diarrhea. I take up myself for cognitive enhancement. I've been taking 250 milligrams a day for over a year, and it's I think it's very helpful. And of course, in the job I have, you need all the cognitive enhancement that you can get your hands on. It's available at New Beginnings, nbnus.com, and other websites. And it's been used at doses up to 2,000 milligrams a day for studies of stroke, brain injury, and Alzheimer's disease. It's been given to animals in which stroke was induced surgically, and then they tried to rescue them at doses equivalent. If it was given in based on human weight, it would be doses as high as 100,000 milligrams a day. So the safety seems very good. And when I looked at, if you look on the WebMD reviews, there was one pretty negative one, but almost all the others were I would say, were pretty positive. Remember, the most important thing, do not confuse phosphatidylcholine. If you hadn't written this down, put phosphatidylcholine and put skull and crossbones over that one. Phosphatidylcholine could be good for you, but not if you have PLA2 and not if you have one of these virus infections. It would probably cause it to be much, much worse. As a matter of fact, that maybe it could kill you. I don't know. But I would definitely not be using that. CDP choline is the inhibitor of the PLA2. So we're in a big fix, probably the scariest time in more than 100 years. And I think it would be very useful to try this out very fast, to put together a fast research study, trying this out on some of the patients who are very ill, maybe on their last legs, and try it out for compassionate use. I would say that since 2,000 milligrams has been used widely for a number of diseases with no harm and mostly big improvements, that it would be worthwhile just to pick 50 patients. And of course, the place to do this would New York City, where all these people are jammed together, and give one group 2,000 milligrams a day, 500 milligrams 4 times a day, which would be 2,000 total a day, and give the other patients placebo, collect the PLA2 at baseline and another one a week later. And if results are good, let the world know about it. Don't wait until a pharmaceutical company approves it. We don't have time to do that. You know, this distancing and quarantining yourself, this may only be able to stop at once, but then as soon as everybody goes off of quarantine, the same thing may happen again. This is such a severe state that, you know, we need to do things differently and not do things as formally. We need to get data fast and using the simplest kind of experiment. So any of you have any of you are physicians or have contact with physicians who are treating large numbers of corona patients, you might want to pass this on. The supplement has been very useful in the treatment of almost every condition for which it's been used and has minor side effects. And since the coronaviruses use PLA2 to enter the cells and replicate, I think another possible use is as to prevent people from becoming infected with the virus to begin with. And to do that, I think you'd have to have a much larger population. So my suggestion would be, again, New York would seem to be New York City would be the ideal place. Find people who are at high risk, like maybe policemen, and have them give half of the group 500 to 1,000 people, give half the group CDP choline and maybe 250 twice a day, and give the others a placebo. And and then maybe in 2 weeks you'd be able to find out if this had any role in preventing new cases. So if we could find something that would prevent the transition, we could prevent the virus and also save the economy. We're at probably the worst place we've been in since the Civil War right now. So some of the other health supplements that can help reduce infection: zinc, 50 milligrams a day. Do not take multiple doses. I would not take more than 50 milligrams a day. And if you take appreciably more than that, you depress your copper absorption so that you can have severe copper deficiency that can result in anemia. And I've seen kids who were given high doses of zinc, they ended up with gray hair when they were 3 years old, because copper is an essential element for hair pigment. Vitamin D may be especially important now because we're at the end of the winter. Most people didn't get some sunshine unless they escaped to a very sunny place over the winter. And vitamin D increases more than 1,000 different immune factors. So that's definitely what we need these days, is very potent immunity. The most common recommendation is 5,000 units a day. I have friends who've taken more than, with multiple sclerosis, who took more than 100,000 units a day. But remember, this is uncharted territory, because excessive amounts of vitamin D can cause your skin to be solidified. So you would be Lot's wife, who turned around and looked at Sodom and Gomorrah and got changed into salt. So if you take too much, that can happen. That almost surely is not going to happen at a dose of 5,000. And most of the physicians I have contact with would be happy with this as an adult dosage. Of course, with children, you might go a little bit lower. Vitamin C, I would say 500 to 1,000. I take the 500. You have to be careful, because very high doses of vitamin C, the vitamin C is converted to oxalates that can cause kidney stones and cause the kidney failure due to the excessive amounts of stones forming in the kidneys. Vitamin A is recommended by the World Health Organization for measles. No exact word about the coronavirus, but for measles, they're recommending 200,000 units of vitamin A for children, and 100,000 units for infants for 2 days to as a prevention for measles infection, because it's found that individuals with vitamin A deficiency are much more likely to develop measles. There is no data for coronaviruses, so this recommendation would be based on similarity. And of course, there's many other immune stimulants out there, like colostrum, transfer factors, and many others, and you might want to check some of those things out as well. The Great Plains wants to develop a new test for immunity, and the biggest problem we've had is getting to validate the test. We want to get people who have already been diagnosed with the coronavirus 19 and had been positive by PCR. So anybody out there can help. We'll put something on the on our website to give the details if you fit this criteria. And we're very nearly ready to launch this test, and this is the last thing we need in order to validate it. And it's going to be very important because it will indicate the degree, how much antibodies you've produced after exposure. It will also help to find out maybe there's thousands of people walking around that have already had it and developed antibodies but haven't gotten sick. So it'd be a tremendous resource to be able to bring to the public. So I would plead with anyone who has this connections or who has had the virus themselves to be willing to give us it would be a blood sample. Thank you. And so thank you very much. And I'm going to take questions for as long as my voice is starting to waver a little bit. So let's get so here is the questions are going to appear here. And I'm going to see, wonder if I can make this larger. I think maybe if I move this, yeah, that does make it larger. Okay, so would melatonin be helpful? Probably. Melatonin is an antioxidant. I think we would. I think we'll post the thing. There's a question about would Great Plains accept samples from other countries? I think that's possible. I have to think it over a little bit. So when we post the thing, we'll put a posting up on the top of our website, like the headline area that gives more information, and we'll answer that. It may take a couple of days to get up. Okay. So the next question is opinion about the efficacy of bleach to kill COVID on contact. I think that's pretty well proven, and there's you can find the exact dilution, and it's because it's a very potent oxidizing agent, and so the oxidizing agent would be that's what I plan using. That's what we're using in our laboratory. So it's ranked with other things, and it's one of the highest ranked things for killing the coronavirus. Okay. I'm sorry, somebody wasn't getting audio. Sepsis, there's a comment that vitamin C has been successful. And I don't know of effects of vitamin C on PLA2. Okay, going on. The slides aren't available, but it will be posted on the website of the Great Plains, so you can go through it again as well. Now a question: does not the level of arachidonic acid push the availability of PLA2 levels in the cell membrane. Push the availability of PLA2. I don't know the answer to that question, but in autism, providing the fish oil, the EPA and DHA significantly reduced PLA2 in the experiment in the children with autism. There's a question, isn't there evidence flavonoids like quercetin help with COVID via inhibition of mast cell inflammation? I don't know about that topic. So far, all the coronaviruses have replicated using PLA2, and I would suspect that information is probably already in the journals, but I haven't found it yet. I would be surprised if it did not, since so far all the other coronaviruses, including the one that was very closely related, the one with the Middle East related virus that was very potent, had very high levels of PLA2. And so I think it's very likely the variety of symptoms, the fact that there is severe breathing damage would also be associated with the high amounts of PLA2 building up in the lungs. And, we could even look, I will bet that there's scientific articles that are probably in press now about the PLA2. Do mycotoxins create PLA2? I suspect they might, but we haven't done the testing. There's lots of Great Plains, of course, does many mycotoxin tests, and it would not surprise me at all if it was some of the damage was associated with induction of PLA2. Question is, can we say emulsifier instead of detergent? Anybody can say what they like to say, of course. I like the word detergent because it brings up into the it creates a mental picture which is exactly correct. These things have exactly the same thing. If you put a solution of Tide into cells, they would cause the same kind of damage that the lysolecithins called. And so, of course, when you give your webinar, you can call it emulsifiers. Am I concerned that professionals will recommend the use of acetaminophen over NSAIDs? Yes, I'm very concerned about it, and especially since, because of this virus, it may be used a lot more. So the autism epidemic began following the Rise syndrome and recommending that acetaminophen began to be used instead of aspirin. And so the same thing could happen with this COVID virus. We could have another autism epidemic if acetaminophen is given. I do not recommend acetaminophen for anybody compared to something like ibuprofen. And there have been medical professionals who have gone out there and said there's absolutely nothing wrong with using ibuprofen instead of Tylenol. And question comes up: are there positive or desirable effects from detergents? I suspect there are. For example, a very positive effect of detergents is human beings. The sperm breaks through the wall of the ovum in all creatures on Earth by producing a phospholipase A2, and that breaks down the cell membrane so that the DNA of the sperm can enter the cell and can enter the egg cell and reproduce a living being. And I'm sure there's many other instances as well. I think the question is, why is there such a variety of animals? I think it's just because the population of China is so large, and the overpopulation led to people eating anything they could get their hands on. I think really that's the reason. You can go right on. There is someone asking about the pangolin theory, and you can get that on Wikipedia. Okay. So does not diet affect the arachidonic acid levels and therefore PLA2 levels? I would say that's true. If you ate a diet that was very high in arachidonic acid, because the lecithins that you're ingesting are very high, it might not cause a direct effect on increasing PLA2, but it might very well if PLA2 were elevated, those foods with high arachidonic acid would produce more toxic detergents. What's different about the CMV mechanism? I would have to review it, but what I recall is that some of the viruses enter the cell and cause the cell to produce excess PLA2. And I think with the CMV, it was the CMV has actually kidnapped the genetic message of the cells to actually produce human phospholipase A2. So the virus is running around with the genetic code of the human inside of it. Would Epstein-Barr virus also use PLA2? I don't know. I mean, there's an awful lot of viruses that do. I did not research. I just researched a good number of viruses, but it was not exhaustive. The children have less PLA2 activity, and could that be why they're less vulnerable to the virus? I mean, that's an interesting idea, but right now I don't remember the exact amount. Is pyrrolidine, the inhibitor of phospholipase A2, a B6 derivative? No, but the name is very similar. So I was very interested if there was a biochemical similarity. I didn't have enough time to do a comparison, but I'll probably be doing that in the next days. What are the natural properties that are beneficial to our body that PLA2 performs before it is hijacked by viruses? It probably, with small increases, it probably is working to remodel the cells. So the cells have a particular structure. PLA2 breaks down some of the structure so that you can think, if you're building a building out of bricks, then that structure is set, but the body is completely fluid. So a small amount of PLA2 might be very helpful to change the structure of the cell to a structure that is more useful for some particular biochemical process. Does ozone therapy play any role in inhibiting PLA2? I'm sorry, I don't have the answer to that. There's a question about resolvents having a positive therapeutic effect on PLA2. And Dr. Bump has a lot of good questions, but I don't know the answer to, and that's one of them. Are there any specific pharmaceutical phospholipase A2 inhibitors recommended? The one that we talked about was the CDP choline, which is used as both a nutritional supplement, but was also used as a pharmaceutical. And I don't know the situation. It may still be sold in Europe as a pharmaceutical with the name Citicoline. Should we be taking the supplement during this time? I think it sounds like a reasonable idea. I'm continuing to take it. If I was infected, I'd be taking the high doses of the CDP choline. But the situation is there is no formal study, but I would like to get one. I think it would be very valuable. But we're in a situation where speed is really important. So if anybody has any ability to look at a study that could be done rapidly using CDP choline on very sick patients or used as a prevention, I would recommend it. How about acetylcholine? To my knowledge, it has not been evaluated as a phospholipase A2 inhibitor. Okay, now a question comes up. They're taking liposomal vitamin C, which the liposomes are made out of lecithin, in an ultrasonic bath. And I have a feeling that the amount of lecithin if people are taking moderate doses, is not going to make a difference. But if people are taking massive amounts of lecithin, this could be problematic if the person has had the exposure to viruses that produce PLA2. I mentioned CMV. It would be this. So I believe that all of these viruses would respond favorably to phospholipase A2 inhibitors. I see Citicoline supplement shown as monosodium salt. That would work fine. So it could be either one. It can say monosodium salt, or it can just say Citicoline or CDP choline. All of these products are equivalent. A person asked, I have a supplement that has 360 milligrams of phosphatidylcholine. So I think this is a relatively small amount, and I'm guessing, I'm not absolutely sure that this small amount would not be problematic. What would be problematic, or a lot of people use infusions of very substantial amounts of lecithin or phosphatidylcholine, and I would think that I would be very wary about doing that during the time of this outbreak. Question, what about people who are taking arachidonic acid supplements for people not converting fats? I'm really not, I'm not sure. I think, arachidonic acid by itself, if it's being used, could be a powerful detergent. Any side effects in healthy individuals? Some mild GI effects. Recommended dose depends on the degree of impairment, what you're using for. And so even small doses of CDP choline reduce the phospholipase in patients with multiple sclerosis. If you have a serious illness, I would use higher doses. Anything that you consider that is really causing a serious illness, I would consider using up to 2,000 milligrams a day. Are you allowed to recommend brand and dosing of citicoline? Really, I've just done it. So citicoline or CDP choline will have some beneficial effects. Most of the time it's sold as in doses of single dose 250 milligrams. And with serious illness, especially if you've had elevated phospholipase A2 in your urine, I think you would consider going to up to 2,000. And some people have used even doses up to 3,000 milligrams a day divided over the day. Okay. There's a question about lecithin sometimes being needed with people who don't have a gallbladder. So one of the functions of the gallbladder is producing bile acids, which are one of the things that have detergent effect. So that's that's one of the positive effects of detergents, are helping to digest food, and I would say I would just be concerned about very high doses of lecithin. A question, would CDP choline help with fibromyalgia? Might it help them? And the phospholipase A2 produces the lysolecithins, which are one of a major pain mediator. So in the sense that CDP choline inhibits the formation of pain, it should be effective in a large number of conditions that are associated with pain, including fibromyalgia. Okay, quite a lot of questions tonight.
Dr. Shah, I can send you the questions tomorrow if you'd like to answer them, and I can send them personally.
Yeah. Okay. Yeah. So yeah, we still have a whole bunch.
Oh, I bet. Yeah. Why don't I just tomorrow email you the questions? You can send me the answers, and I will email the people that ask them the answers.
So you mean that I just don't want to have to write an email for every one of these.
No, you don't have to. I will.
Oh, okay. Okay. Yeah. That sounds good. So we
Okay,
have,
so if your question wasn't answered, then it will be.
yeah. Okay. So, so you can put me back on and I'll say good night.
You're on.
Okay. So, I think my voice is, giving out and so I'm going to call it a night, and we'll tomorrow attempt to answer. We probably only got through about a third or a fourth of all the questions that were given. So we'll make an attempt to answer a lot more of them tomorrow. Thank you all, and God bless.